welcome to today's video lecture. In today's video lecture, I'll be solving previous year's NEET questions and discussing the answers. And this is for the chapter Biotechnology, Principles and Processes. Okay, and we are in part 1 of the series, right? So let's begin. Okay, so question number 1. The DNA molecule to which the gene of interest is integrated for cloning is called, this appeared in the year 2015, A. template, B. carrier, C. transformer, or D. vector. Okay, so the DNA molecule to which the gene of interest is integrated for cloning is called D. vector. Alright, so the correct answer is D. Let's go to the discussion. Okay, so here you see a diagram of a vector, a plasmid vector. It's called PBR322. Okay, it's an artificially created cloning vector and it's a plasmid. Okay, so vectors are DNA molecules which can carry a foreign DNA to be cloned. All right, so you've studied human health and diseases, right? So mosquitoes behave as a vector for plasmodium, right? The parasite, the protozoa that causes malaria. Okay, similarly over here, it's the same concept. Vector is like a vehicle. It just carries the DNA, the foreign DNA that we want to be cloned. Okay, so similarly, this is like a DNA molecule, okay, into which the foreign DNA is inserted, okay, and this can then be put into a host cell, which then produces the protein that we want. Okay, so this plasmid that you see here is called the vector. Okay, so they can replicate by themselves, and there are different types of vectors that are available. One of them is plasmids, then there's bacteriophage, there's cosmids, and there are artificial chromosomes also. Okay, so plasmids usually are found naturally in bacteria. So they are used as vectors, but now over here in genetic engineering there are artificially created plasmids over here, and into which a gene of interest can be inserted into and then can be cloned, okay, for your interest. So yeah, it is vectors. If you go back to the question, the DNA molecule to which the gene of interest is integrated for cloning is called vector. Okay, let's move on then. This was pretty simple. Question number 2. The cutting of DNA at specific locations became possible with the discovery of, and this appeared in the year 2015 again, A. selectable markers, B. ligases, C. restriction enzymes, or D. probes. Okay, so the cutting of DNA at specific locations became possible with the discovery of restriction enzymes. Okay, and the answer is C. Let's go to the discussion. So, yeah, restriction endonuclease or restriction enzymes have the ability to cut or split the DNA into different segments, okay, and they are done at specific sites. Okay, so they were first discovered in E. coli, where they were used to cut bacteriophages, thus hindering their replication. So restriction enzymes in E. coli were used as a defense mechanism for cutting the bacteriophage that attacks them. Okay, so once this was discovered, these restriction enzymes, they realized that these restriction enzymes can be cut at specific locations on the DNA fragment. Okay, they found out those specific locations and they used those restriction enzymes. They isolated those restriction enzymes from the bacteria, and then they were used to cut the DNA at those specific sites. All right. So for example, EcoR1. Okay, so they are named as EcoR1 like based on the genus and species. Then R is actually RY13, which is a strain, I'm sorry, and 1 is order of discovery. Okay, so EcoR1, then there is Hind3. Okay, Haemophilus influenzae is the genus and species, RD is the strain, and 3 is the order of discovery. Okay, so like that there are various restriction enzymes that have been isolated and they have been found to cut at specific sites on the DNA fragment. Okay, clear? And one more thing, yeah, they cut, and we'll probably come to this later on as we move on with the questions, is that they cut at specific sites that are palindromic. Okay, that means if it reads from left to right, it reads from right to left the same way. Okay, those are palindromic nucleotide sequences. All right. Okay, this was also again pretty simple. Let's move on then. Question number 3. Which one of the following is a case of wrong matching? Okay, this is wrong matching. This appeared in the year 2012. A. Somatic hybridization fusion of two diverse cells. B. Vector DNA site for tRNA synthesis. C. Micropropagation in vitro production of plants in large numbers. D. Callus, unorganized mass of cells produced in tissue culture. So the wrong matching is B. Vector DNA is not the site for tRNA synthesis. Okay, so the answer is B. We already discussed vector DNA, so it's pretty much the same answer. They carry a foreign DNA to be cloned. Okay, and they can replicate by themselves. Yes, they have the ability to do that. And some of the examples are plasmids, bacteriophage, cosmids, and artificial chromosomes. So this is one of the features of a vector. They should be able to replicate themselves. Okay, clear? Let's move on then. Alright, so question number 4. Which one of the following techniques made it possible to genetically engineer living organisms? And this appeared in their mains 2011. A. Recombinant DNA techniques. B. X-ray diffraction. C. Heavier isotope labeling. Or D. Hybridization. Okay, so clearly it is A. Recombinant DNA techniques. Alright, so the answer is A. Let's go to the discussion. So what are the steps that are applied in genetic engineering? Okay, so recombinant DNA techniques, they assist in the genetic engineering of living organisms, like inserting a foreign gene into a vector and then putting it into a host. You know, all these, there are different steps required, right? So first is identifying the gene of interest, then isolating it, and then identifying the ideal vector for it, and then identifying the ideal host for it, so that the eventually the protein is produced. And we would usually require the protein to be produced in large amounts. Okay, so that's bioprocess engineering, which is the downstream process. And eventually that is what we want the product, which is the protein. Okay, after cloning the vectors. Okay, so this is how we create the recombinant DNA and then eventually get the protein that we want. Okay, so first we need to isolate the desired gene fragment. Okay, so you have this let's say you have this whole DNA, this whole structure over here. So we identify the gene that we want. Okay, so this particular say gene fragment codes for a particular protein. Okay, we have established that. Okay, so we isolate that gene fragment. Okay, that's first step. Then we select the appropriate vector DNA. So for example, let's say for this particular gene fragment, let's say the PBR322 is the ideal gene, the ideal vector for that. Let's assume, okay, because there are different gene fragments, different lengths of the gene fragments. So not all gene fragments can be used in PBR322. You have to find the appropriate vector for that. So if there are bigger gene fragments, you can use cosmid or artificial chromosomes, used like that. So let's say for this case, this particular gene fragment codes for a particular protein, and we can use the vector PBR322, which is ideal for us. Okay, now insert the desired gene into the vector DNA, forming our DNA. Okay, so you've identified the gene, you've identified the particular vector that can be used. So let's say you have PBR322 and you use the restriction enzyme to cut this particular fragment of the vector, and you already have your, let's say I'll use a different color, you already have your isolated gene fragment over here. Okay, let's get rid of this. So now you have space created over here for this gene fragment to enter. Okay, you have space created here for this gene fragment to be placed here. All right, so you place it over here. If you need to use DNA ligase, yes, you need to use DNA ligase to attach both the ends of the that sticky end or whatever blunt end that is created from the restriction endonuclease, restriction enzymes, as well as the vector. Okay, and you attach the ends. Okay, once that end is attached, now it becomes part of the vector. Okay, so you have an ORI, which is origin of replication. Okay, which will then begin to replicate this vector, that means clone these vectors, and so you'll have many plasmids that now contain the new gene of interest, and these gene of interest will then produce the protein that you want. So basically that is what it is. Now when this new gene of interest is attached over here, okay, this is called recombinant DNA. Okay, once this gene of interest has come and attached over here, this is called recombinant DNA. All right, so basically that is where the term applies to. So now you introduce the rDNA into the host cells. Okay, let's say E. coli is the ideal host cell for this rDNA. Okay, so you introduce into the host cells by the process of transformation. Okay, transformation. All right, and then multiplication and selection of clones containing the rDNA molecules and the expression of gene to produce desired product. Okay, so once the DNA is introduced, the rDNA is introduced into the host cell, that means the plasmid which contains the new gene of interest that we want is introduced into the host cells. Now plasmids have the ability to replicate, right? The vector has the ability to replicate on its own, so it begins to replicate on its own and produce many copies. Okay, many copies of that particular vector. Okay, and now once that is there, you need to select which ones are containing the new. Now when you insert the rDNA into the bacteria, let's say E. coli, okay, it's not necessary then that all the cells will be transformed, okay, containing the new rDNA. Okay, there'll be some E. coli cells which will not have that. That's why you have selectable markers on a plasmid. If you go back and read NCERT, you'll notice that every vector has a selectable marker which helps us to easily identify which E. coli have the transformant, have the rDNA molecule in it, and which ones don't have it. All right, so obviously, yeah, then you need to perform a test where you need to identify the cells which contain the transformed, that means the new rDNA molecule, which ones don't have it. Once you do that, you can then produce the desired product from that, because we don't want the cells which don't have the rDNA. That is not of our interest, right? We only want the cells which contain the rDNA. Okay, so these are the different steps that are required to form the that are used in genetic engineering to produce the rDNA and then eventually produce the product that we want. Okay, as we go down further, you'll be able to understand this better. If this is all new to you, it's difficult to understand. Yes, it's a pretty new science that is applied now. So yeah, let's move on to the next question then. If we go back to our question, I've answered the question. Okay, so question number 5. Which of the following are used in gene cloning? Okay, this appeared in the year 2010. So the answer is D, plasmids. Okay, none of the other options make sense. So the answer is D, plasmids. So plasmids are used in gene cloning and they are extra chromosomal, double stranded, circular, self-replicating DNA. So now almost all bacteria contain plasmids and have a low copy number or high copy number. Copy number is the amount of copies that it can produce. They can vary from 1 to 20 or they can go up to 100. Okay, so that obviously depends on the original origin of replication, okay, in the plasmid, and the size varies from 1 to 500 kilobase. Okay, so this was pretty simple actually. Which of the following are used in gene cloning? It is plasmids. Okay, question number 6. Manipulation of DNA genetic engineering became possible due to the discovery of, and this appeared in 2002, A. Restriction endonuclease, B. DNA ligase, C. Transcriptase, or D. Primase. Okay, so manipulation of DNA became possible due to discovery of restriction endonuclease. So with this discovery, we could insert a foreign gene into a vector. Okay, that was possible. So restriction endonuclease has created an ability to form rDNA clones, like I previously explained in the steps in genetic engineering. Okay, there are specific sites that it was able to identify, and scientists discovered that, and they were able to use it to insert foreign genes into a vector, let's say a plasmid. Okay, and they were able to clone that plasmid. So restriction endonuclease, they have the ability to cut or split the DNA at specific points on both the desired gene as well as the vector DNA. And this is a very important information, that when you are using restriction endonuclease, when you make a cut in the DNA fragment that you want, the same restriction endonuclease has to make a cut in the vector DNA. Okay, only then you'll have complementary nucleotides. Okay, and then you'll have not all restriction endonuclease will produce a sticky end cut. There can be some which will produce blunt ends, so there won't be any complementary DNAs there. Sorry, complementary nucleotides there. But yeah, it is always preferable to use the same restriction endonuclease to make a cut in this DNA fragment that you need the gene of interest, and to make the same cut in this vector DNA to have the complementary nucleotide, so that this DNA as in this gene fragment can be inserted into this vector DNA. Okay, maybe later on, I feel like there should be another video lecture only explaining the steps on genetic engineering. That would be very helpful, because this is just in bits and pieces. So if you have read NCERT properly, this should make sense to you. If you haven't, then it will be very difficult for you to understand. Okay, so I would suggest that you go back and read the chapter in NCERT and then come back to this, because I'm only explaining bits and pieces in the middle. So yeah, thus creating an opportunity to produce rDNA, right? So restriction endonuclease played a very important role in creating the rDNA, the recombinant DNA. All right, let's move on. Question number 7. The bacteria generally used for genetic engineering is, this appeared in the year 2000, A. Agrobacterium, B. Bacillus, C. Pseudomonas, D. Clostridium. So the answer is A. Agrobacterium. Okay, answer is A. So Agrobacterium tumefaciens, okay, it's actually a pathogen that affects dicot plants, and it produces a tumor. Okay, they transfer the tDNA. Okay, so there is a vector, a plasmid inside, which contains the tDNA gene. Okay, now this tDNA produces tumors in plants. Okay, now these tumors are known as crown galls. Okay, these tumors are known as crown galls. Okay, and the plasmid is Ti plasmid. It's tumor inducing plasmid. Okay, but it induces tumor formation. So now what scientists did is, they thought that if we remove this tDNA, then it won't be pathogenic anymore. Yeah, the tDNA is what's causing the tumor in these plants, right? So if you remove the tDNA, then it won't be pathogenic anymore. In that place, what we can do is put another gene of interest that we want. Okay, that would be helpful for the plant. Okay, so this plasmid has been modified, which is not pathogenic and is used as a natural genetic engineering of plants. Okay, so this tDNA was removed, and in that place they put another gene of interest, and then they introduced it into the plants. Since this bacteria easily becomes a part of the genotype of the plant. Okay, so it becomes a part of the plant, so we can put our gene of interest in it and then allow the plant to grow accordingly. Okay, so this was one of the ways. This is natural genetic engineer. Okay, because the Actobacterium tumefaciens, easily it does affect the dicot plants, but we don't want the tumor part in it. Okay, that is going to reduce the plant, into just a tumor formation and crown galls. We don't want that. Okay, so instead we put our gene of interest in it and then allow it to grow. Okay, so yeah, the bacteria generally used for genetic engineering is agrobacterium. All right, that's the answer. That's the direct answer to your question. But why was it like that? Because of this reason. Okay, let's move on then. Question number 8. Which of the following is related to genetic engineering? This appeared in the year 99. A. Heterosis, B. Mutation, C. Plastid, and D. Plasmid. So, which of the following is related to genetic engineering? It is D. Plasmid. So, the answer is B. I think we already discussed plasmid before, so I don't want to go much into it again. Yeah. So it's basically the same thing. I've just added the different ranges that high copy number or low copy number have. It can be the 1 to 4 plus per cell, or 10 to 100 per cell. All right. Let's move on then. Question number 9. Genetic engineering is possible because, and this appeared in the year 98. A. We can cut DNA at specific sites by endonucleases like DNase 1. B. Restriction endonuclease purified from bacteria can be used in vitro. C. The phenomenon of transduction in bacteria is well understood. D. We can see DNA by electron microscope. Okay, so genetic engineering is possible because we already discussed this. B. Restriction endonuclease purified from bacteria can be used in vitro. So this was a very major discovery that was made. Okay, so the answer is B. The restriction enzymes are purified from bacteria and are used in vitro. They cut at specific sites on the DNA fragment, but DNase 1 is non-specific and cleaves DNA at phosphodiester cleavages. So option A was incorrect, okay, because DNase 1 is not really what we need. DNase 1 is usually used for RNA purification, in labs when you want to purify RNA and want to get rid of the DNA. So DNase 1 is used, and it denatures DNA by cleaving at the phosphodiester linkages. Okay, but if you look at the X restriction enzyme, the discovery was very important because now we could isolate our gene of interest. We could use the vector in our favor. Okay, so that way we could genetically engineer, okay, these cells to produce the protein of our interest. Okay, this pretty much answers our question. Yeah. All right, let's move on. Question number 10. When scientists make an animal superior by view of genotype, introducing some foreign genes in it is called. This appeared in 96. A. Immunization, B. Genetic engineering, C. Tissue culture, D. Biotechnology. So when you are introducing some foreign genes in it, it is called B. Genetic engineering. Okay, so the answer is B. So genetic engineering, and this definition is according to NCRT. Okay, genetic engineering is the technique to alter the chemistry of genetic material, that is DNA and RNA, to introduce these into host organisms and change the phenotype of the host organism. That means it begins to produce a particular kind of protein that we want. So when you introduce, we already got into the details of genetic, and this is a very generic statement, But when you begin to understand the steps that are involved in genetic engineering, that's why I would suggest you read NCRT before you come here and follow the questions. There are different steps and you have to follow them in order. Okay, otherwise it will not make sense to you. So you need to isolate the gene fragment, you need to find the appropriate vector, you need to use the same restriction endonuclease in both the isolating the gene fragment as well as the vector, then use ligase to bind the fragment, and then put it into an appropriate host, for example E. coli transformation, that is, and then select the cells that have the rDNA in it, and then you can use that to produce the proteins. Okay, so yeah, basically these are the steps that are involved in genetic engineering. And biotechnology, genetic engineering forms a part of biotechnology. There's genetic engineering as well as bioprocess engineering. Okay, these are the two steps in biotechnology. Okay, so but according to the question, when you are introducing some foreign genes in it, it's called genetic engineering. Okay, all right, let's move on then. Question number 11. Which of the following organelles is related with genetic engineering? This appeared in 94. A. Mitochondria. B. Plasmids. C. Golgi bodies. D. Lysosomes. Okay, so clearly it is plasmids. We already discussed this. The answer is B. So this is very important, I guess, because they keep asking this question over and over again. So plasmids, again, I'm not going to repeat myself again. You can pause the video and go through the steps and the answers here, the discussion. And I guess we'll move on to the next question. Alright, so question number 12. Identify the wrong statement with regard to restriction enzymes. This appeared in the year NEET 2020. A. Each restriction enzyme functions by inspecting the length of a DNA sequence. B. They cut the strand of DNA at palindromic sites. C. They are useful in genetic engineering. D. Sticky ends can be joined by using DNA ligases. Okay, so identify the wrong statement. So A is correct. They cut the DNA at palindromic sites. Yes, we know that. There are specific palindromic sites too. C. They are useful in genetic engineering. Yes. Sticky ends can be joined by using DNA ligases. Now that is a little that is incorrect. So the answer is B. Okay, so now there are two different types of ends that can be formed after the restriction endonuclease has acted on the gene fragment. Now you can get either now what you see here, HAE3, either a blunt end. Okay, so let's say this is a palindromic site that HAE3 identifies. Okay, and it cuts in the middle. Okay, so you get GGCC over here, CCGG over here. Okay, so these are blunt ends. These are called blunt ends. Alright, so blunt ends formed by restriction enzymes, cuts are joined by DNA ligase. Now you don't need, there is no complementary nucleotide or anything required. So the DNA ligase, let's say you have a blunt end formed by the vector and you have this blunt end formed by the restriction endonuclease. Okay, so you have this gene fragment over here, you have this vector DNA space over here. Directly go and stick it in. Okay, the DNA ligase, that's the job. Okay, it sticks, it inserts that DNA and sticks the ends there. Okay, phosphodiester linkages are formed. Okay, over here. So DNA ligase does the job when it comes to blunt ends. Now you understood the concept of what blunt ends are, right? So that is one example that I've given here is HAE3. Okay, this particular restriction enzyme, if you can remember it, is good. They might ask you in the exam. I don't know, but it would be nice to remember. Okay, produces blunt ends. Now, in the case of sticky ends, both the DNA fragment of interest as well as the vector need to be cut by the same restriction enzyme to allow binding of the gene of interest and the vector. Okay, so that means both, I think I have an example here. I think it would be better to show you. Yeah, this is a very good example, eco R1. Okay, you have this palindromic site here, GAATTC and GAATTC. Eco R1 identifies this particular sequence. Okay, so it will cut here, it will make a cut here. Okay, the phosphodiester linkages are broken in these parts. Okay, so the product that you get is this. That means it's going to cut over here. This is what you get. This is the first product and this is the second product. Sorry. Okay, you get these two fragments over here. Alright, so these are called sticky ends. Okay, that's the difference between blunt ends and sticky ends. Alright, so over here now both need to be cut using the that means the vector DNA. That means you have your gene of interest and both need to be cut with eco R1. Okay, now you have the vector, both need to be cut with eco R1 to produce the complementary nucleotide. Otherwise it will not make sense, right? If you have eco R1 cut, as in you have another particular nucleotide, I'm sorry, restriction endonuclease cut, I mean to say you have eco R1 cut by a particular genome fragment, and then you have another restriction endonuclease cut by another vector, then you're going to produce those complementary nucleotides. So you need to use the same endonuclease in both, in the foreign gene that you want as well as the vector gene, the vector DNA. Okay, so that it produces those complementary nucleotides. Now once those splits have occurred in the vector DNA as well as the gene of interest, it is easy for them to stick. For then you don't need DNA ligase. Okay, you don't need DNA ligase for sticky ends that are cut by both the restriction endonuclease, that means the gene of interest as well as the vector DNA. Okay. So in the case of sticky ends, both the DNA fragment of interest as well as the vector DNA need to be cut by the same restriction enzymes to allow binding of the gene of interest and the vector. Now, if that is done, only then the sticky ends will stick. You don't need DNA ligase for that. So sticky ends can be joined by using DNA ligases. It's not really true, because with sticky ends and you're using both restriction endonuclease in the gene of interest as well as the vector DNA, you will get complementary nucleotide to easily bind and form those linkages. Okay, clear. But for blunt ends, yes, blunt ends DNA ligase does a very good job in attaching the gene of interest into it. Okay, so question number 13. Choose the correct pair from the following. So we are in NEET 2020. A ligases join the two DNA molecules. B polymerases break the DNA into fragments. C nucleases separate the two strands of DNA. D exonucleases make cuts at specific position within DNA. So the correct pairing is A, ligases join the two DNA molecules. The others are not true. Answer is A. So DNA ligase joins the DNA fragments at the phosphodiester linkages. They were originally isolated from viruses and they also occur in E. coli and eukaryotic cells. Okay, so you see over here, this is B represents base. This is the phosphodiester linkage. This looks like a blunt end. Okay, so if DNA ligase is there, it will attach. Okay, clear? Alright, so DNA ligase does the job of binding the two DNA fragments together. Basically, that's what its job is. Okay, let's move on then. Question number 14. The specific palindromic sequence which is recognized by ECO R1 is, and this appeared in NEET 2020, A GAATTC GGAACC CTTAG GGATCC. Okay, so the specific palindromic sequence is by ECO R1, which is GAATTC. Okay, the answer is A. This is there in NCERT, so I would suggest you remember this sequence. The specific palindromic sequence recognized by ECO R1 is this. Please remember this palindromic sequence for ECO R1. Alright, and this is there in NCERT, so that's why it's been asked. Alright. Okay, this was pretty direct. Question number 15. The sequence that controls the copy number of the linked DNA in the vector is termed, this appeared in NEET 2020, A. Selectable marker, B. ORI site, C. Palindromic sequence, D. Recognition site. Okay, so the sequence that controls the copy number is ORI site. Answer is B. Alright, so here you have a plasmid, right? You have an origin of replication. Now this origin of replication is what controls the copy number in the plasmid. So there are different types of origin of replication which have certain types of copy numbers that it can replicate the plasmid into. Alright. So the vector present in the host cell has a copy number which represents the number of copies present in the cell. The foreign DNA along with the vector form the rDNA, which will undergo replication with the help of ORI. So let's say a foreign DNA was inserted here. Okay, so now this copy number that is produced by this ORI depends, the ORI basically determines the copy number of the vector. Okay, a foreign DNA is inserted here and the rDNA is produced. Right, the rDNA is produced and this will then replicate based on this origin of replication. So this origin of replication, now we are not going to the details of how it is regulated, we're not going to the genetics of that, but just understand that the origin of replication has the ability to control the number of copies that the plasmid can be replicated into. We don't, sometimes there are instances that we don't want so many copy numbers, or there are instances that we want many copy numbers. So there are different types of origin of replication that can be inserted in the plasmid when it's created, or the vector when it's created, to control how many copies that we can produce. So if the rDNA is attached to it, as in the foreign gene is attached to it to produce the rDNA, then we can control how many copies of that gene of interest that we want, and that is based on the ORI. Okay, the origin of replication. Okay, so if you go back to the question, this is actually a good question. Okay, the sequence that controls the copy number of linked DNA in the vector is termed BORI site. Now this this information is there in NCERT, but it's not really understood what copy number is and how the ORI site determines that copy number. Okay, so the ORI site are of different types. There are different types of ORIs and they have a different copy number that they can replicate the vector into. Okay, so in that way, in turn, eventually the rDNA that we have formed, we can control the number of copies that are produced. All right, clear? Question number 16. In gel electrophoresis, separated DNA fragments can be visualized with the help of, this appeared in NEET 2020, A. acetocarmine in bright blue light, B. ethidium bromide in UV radiation, C. acetocarmine in UV radiation, D. ethidium bromide in infrared radiation. So it is B. ethidium bromide in UV radiation. Answer is B. So yeah, this diagram is from NCERT. This shows a gel electrophoresis technique. These are the wells in which the DNA samples are put in. Okay, this is the cathode side. This is the cathode side. This is the anode side. Okay, and the DNA will travel from the cathode to the anode side under a electrical field. All right. Now the gel, it's called gel electrophoresis, right? It's called agarose gel. Okay, agarose gel is used to prepare the agarose gel, pour it into this well-like structure, okay, in the lab, and you will have to enter or place your DNA samples in it. Okay, basically that's what the system is, the technique is. Okay, so gel electrophoresis technique by which DNA after treatment with restriction enzymes can be separated. The samples are placed in wells with the help of electric charge. I already explained that. And when these samples move down, they are separated based on their size. Okay, that's very important. Now this agarose gel that is used here have sieves in it. Okay, you know, like a sieve like structure you've seen the filter at home that you use, there are holes in it. Okay, so this similarly, this also have, you know, like a sieve like, you know, structure, nature to this gel. And so when these DNA samples are moved down the wells, they separate based on their sample, the size. Okay, so the largest is on top, the smallest is in the bottom. Okay, the largest on the top, smallest is in the bottom. Now, so for example, you have cut the DNA fragments into using restriction endonuclease into various. Now they split into different types of sizes. We don't know that. We just have a DNA structure here. We've entered the restriction endonuclease and we've got different cuts. Okay, that means that of different fragments of different lengths, but we don't know what it is. Now to see that, we have to run a gel electrophoresis method. So okay, so when you enter the sample inside, the samples are then divided based on their length. That means the largest stays on the top, the smallest goes down to the bottom, and then you can view them. Okay, once the separation is done by gel electrophoresis, the separation is done by gel electrophoresis, and you can view them by adding a fluorescent dye. Okay, and you can view them under UV radiation. Okay, the samples are placed in wells with the help of electric charge, and the samples move down the gel and separate based on size. The large size fragments remain at the top, whereas small size fragments move to the bottom. Okay, I've just explained that. And they can be viewed when treated with ethidium bromide. Now, ethidium bromide is actually a fluorescent dye, and then when placed under a UV light, you can see it glow. Now, I don't have a real picture of it, unfortunately, but basically when you view them, you can take a picture of your gel, so you know exactly, you know, what sizes they are. This basically a marker. One of the wells have a marker, so you can compare. Okay, this is a marker of different base lengths, and you have your sample. Okay, and this will then have different sizes, as in different lengths of the fragments that have been separated, and you can compare it with the marker and see where does your sample lie. Okay, so that's how it works. Yeah, this is theoretically, and I know it's difficult to understand unless until you know actually see it, but we can't really do anything about that. We have to understand it theoretically over here, but just imagine it, or if you can watch a YouTube video or something on it where they have explained, where they have shown how gel electrophoresis works. I'm sure there are many resources available now and you can visualize it. But once you have seen it practically, you'll be able to understand how it actually works. Okay, but to answer your question, it's pretty simple. Theoretically, if you have to just, you know, study for the exam, then yeah, ethidium bromide in UV radiation is the right answer. All right, let's move on. Question number 17. Following statements describe the characteristics of the enzyme restriction endonuclease. Identify the incorrect statement. This appeared in the year NEET 2019. So you identify the incorrect statement. A. The enzyme recognizes specific palindromic nucleotide sequence in the DNA. B. The enzyme cuts DNA molecule at identified positions within the DNA. C. The enzyme binds DNA at specific sites and cuts only one of the two strands. D. The enzyme cuts the sugar phosphate backbone at specific sites on each strand. The incorrect statement is actually C. It does not cut only one of the two strands, it cuts both the strands. So the answer is C. So the enzyme binds to both strands of DNA, cuts them at specific sites. Now you can see here from the case of EcoR1, it will cut here as well as here. It won't just cut over here and not here. Okay, so it cuts at both the sides. All right. So it was pretty simple. Okay, so question number 18. A selectable marker is used to, this appear in NEET 2019, A. Help in eliminating the non-transformants so that the transformants can be regenerated. B. Identify the gene for a desired trait in an alien organism. C. Select a suitable vector for transformation in a specific crop. D. Mark a gene on a chromosome for isolation using restriction enzyme. So selectable marker is used to help in eliminating the non-transformants so that the transformants can be regenerated. The answer is A. Now, one of the quality that a vector should have is to have a selectable marker. Okay, in the case of PBR 322, it has tetracycline resistant gene and an ampicillin resistant gene. Okay, now we already discussed the steps of genetic engineering. Okay, let's say you already have a rDNA molecule vector ready. Okay, you have your gene of interest over here. This yellow is the gene of interest. You have that prepared. Okay, this is your rDNA molecule now. Now this rDNA molecule particularly is placed in the tetracycline resistant gene. Let's see. Okay, you have used BAM-H1. You have used BAM-H1 to isolate your gene fragment. You have used BAM-H1 restriction endonuclease to create a space here space here in the vector DNA, and you have inserted the foreign DNA into this region. Okay, so now you have rDNA ready. Okay, now you have rDNA ready and you are now put it into the E. coli host. E. coli host. Okay, you have your culture of E. coli host and you have transformed the cells, E. coli cells, and placed it in. Now some of these E. coli cells will contain that, some of them will not. Okay, some of them will contain the rDNA and some of them will just contain a normal plasmid. Okay, so we don't know which one is what, but we do know that we have created a cut in the tetracycline resistant part of the gene. So that means this gene is now inactive, whereas amplicillin resistant gene is still active. We know that for sure. Okay, now this is inactive. Let's say I just put a cross here and this is active. That means when we culture a plate, a petri dish, you put the medium in this petri dish plate and you culture this E. coli cells here, allow them to grow. Okay, and this particular medium contains ampicillin, for example. Okay, now this medium contains the antibiotic ampicillin. Okay, you now culture the E. coli, all that E. coli which you've transformed all the E. coli cells. Now you know that some of them have it, some of them don't. Okay, practically this is what it is. Now you should be able to select the transformed from the non-transformed cells. Okay, you culture a plate containing a medium containing ampicillin in it. But now we know that the ampicillin resistant gene is active in transformed cells. Yeah, so they will grow here. All the plasmids that contain rDNA will grow here. But we want the ones that are inactive, tetracycline resistant gene. These are the ones that contain that. We don't want all the ones that have the ampicillin resistant gene. So we make a replica. of the same plate. Replica means now you have, let's say, practically if you look at it, if you are plating a dish, and this all you'll study if you study microbiology, if you are plating a dish, you will place this colonies over here. You're making the exact replica over here. Okay, making the exact replica over here. Let's say you have a tetracycline plate. Okay, and you make the exact replica over here. Now some of them will grow, some of them will not grow. Okay, let's say some of them will grow, some of them will not grow. Okay, so now what happens? That means these contain the tetracycline resistant gene. The ones that don't grow are the ones that have the inactive gene. Okay, you compare both the plates. Now you can choose the transformed colonies. Now you can choose the transformed colonies. All right, so this contains ampicillin. All of them will grow because they are resistant to ampicillin. But in tetracycline, not all of them will grow. The ones that have the active gene only will grow. The ones that don't have the active gene will not grow. We can see now, compare. It has to be a replica. It has to be a replica. That means you have a colony here, you should have a colony here. You can have a colony here, you should have a colony here, and so on. Okay, now you see over here it's growing, but here it's not growing. That means this is the recombinant gene. Okay, so that's how a recombinant DNA. Okay, so these are the colonies that have the recombinant DNA because they did not grow in tetracycline resistant, a tetracycline medium plate. That means the tetracycline resistant gene is inactive in there. So that's how you know that these are the colonies, or these are the E. coli cells that have the foreign gene inserted. That is why in a vector it is important to have a selectable marker. Okay, yeah, again this is all practical knowledge, but you have to learn this theoretically. That's how it's actually done in a lab, and that's how they are able to select transformants from non-transformants. All right, so again I'll repeat myself. You have ampicillin resistant gene, you have tetracycline resistant gene. Okay, in the case of we know that the foreign gene is inserted in the tetracycline resistant part of the vector. Okay, so now this is inactive. That means it's no longer resistant to tetracycline. We know that for sure. So we first plate ampicillin resistant petri dish that contains ampicillin, but it is resistant to ampicillin. We know the E. coli colonies are resistant to ampicillin. So it will eventually grow, all the colonies will grow. And we create another replica with a medium in the petri dish containing tetracycline. Now we can compare both and see where in which position have the tetracycline resistant colonies not grown. That means the tetracycline gene is inactive in those, and that contains the foreign gene of interest, the foreign DNA that we want, the recombinant DNA molecules that we want. Okay, so that's how it is important to have a selectable marker. All right, and that's how it's actually done in a lab, to be able to select the transformants from the non-transformants. Okay, I hope you've understood this. And we've already theoretically, if you have to remember this, a selectable marker is used to help in eliminating the non-transformants so that the transformants can be regenerated. Okay, so that pretty much answers our question. All right, question number 19. Given below are 4 statements pertaining to separation of DNA fragments using gel electrophoresis. Identify the incorrect statements. One, DNA is negatively charged and so it is loaded on gel towards the anode terminal. 2, DNA fragments travel along the surface of the gel whose concentration does not affect the movement of DNA. 3, smaller the size of DNA fragment, larger the distance it travels through it. 4, pure DNA can be visualized directly by exposing UV radiation. To find the incorrect statements. So DNA is negatively charged, so it is loaded on gel towards the anode terminal. Actually, it's loaded on gel, sorry, at the cathode terminal towards the anode terminal. I don't understand. This framing is a little problematic, but I would assume that they're loaded on gel at the cathode terminal, not at the anode terminal. So, one is incorrect. Sorry, DNA fragments travel along the surface of the gel whose concentration does not affect movement of DNA. Concentration does affect the movement of DNA. So 2 is also incorrect. 3, smaller the size of DNA fragment, larger the distance travels through it. Yes, that's correct. Pure DNA can be visualized directly by exposing UV radiation. No, that's incorrect. You need a fluorescent dye for it. So 4 is incorrect. So 1, 2 and 4. So the answer is D. This appeared in NEET 2019. So the answer is D over here. Let's go. So DNA is negatively charged. So it will be loaded in the cathode side of the gel. The DNA fragments that travel along the gel are affected by its concentration. Now we can't have too much concentration of the DNA when we are loading on the gel. Okay, now there are various experiments that have been done to identify the correct concentration of the gel that we need to run it. Correct DNA concentration we need to run it on a gel. Okay, the agar gel during a gel electrophoresis. So, for example, if you have too much concentration of the DNA, the increasing concentration of DNA affects the size of the fragment. Okay, it has been found that it affects the size of the fragment, thus resulting in misidentification of genes. So if you concentrate the DNA too much and you add it into the gel, and you want to find the correct base pair that it is, that is the length of the fragment, but you will not be able to find it if it is very concentrated. So an ideal concentration is required. Okay, you can't have 800 micrograms or anything like that, it has to be in approximately of 80 micrograms, so on. But there are various experiments that have been done, okay, to suggest that the concentration of DNA plays an important role when running the gel electrophoresis. The DNA samples are stained with and this is an important piece of information, please remember this. Okay, the DNA samples are stained with fluorescent dyes and exposed to UV for visualization. Ethidium bromide is a fluorescent dye, which is it is stained. The DNA samples are stained with that, and then when it's exposed to UV radiation, you can visualize your fragments, the DNA fragments. Okay, so yeah, pretty much answered the question here. Yeah, 1, 2 and 4 are incorrect. This was a good question. Now, biotechnology is usually a practical science. So, you are instead learning it in theory. So, it's a little difficult to understand the theory part of it until you see it or do it practically. But yeah, at school level, unless until you pursue research later on, that's a different thing. You'll eventually do it practically and you'll understand it better. But from the exam point of view, I would just suggest you read the chapter properly and follow the steps that are involved. Try and visualize it as it's done, and then maybe that will help you remember the steps. Again, help you understand the steps better. Okay, so question number 20. Which of the following is commonly used as a vector for introducing a DNA fragment in human lymphocytes? Okay, this appeared in NEET 2018. A. Retrovirus, B. TI plasmid, C. Lambda-phage, D. PBR322. So retrovirus, the answer is A, is used as a vector for introducing a DNA fragment in human lymphocytes. Okay, so the answer is A. Like previously, we spoke about TI plasmid and how that is used as a natural genetic engineer for plants. Similarly, retroviruses are used in animals. Okay. So the ability to transform normal cells into cancerous cells, we know that. So the virulent part is removed. Okay, they behave as useful vectors to transfer genes of interest to humans. And in the case of the question, it says human lymphocytes. So yeah. So retroviruses have the ability to do that, and they also a form of natural genetic engineer, just like TI plasmids. Okay, remember this as a fact. Question number 21. The DNA fragments separated on an agarose gel can be visualized after staining with, this appeared in NEET 2017. A. Acetocarmine, B. Aniline blues, C. Ethidium bromide, D. Bromophenol blue. So we already discussed this, C. Ethidium bromide. Answer is C. Okay. So ethidium bromide is a fluorescent dye which can help visualize DNA when it is exposed to UV light. Okay, and it's a way to see where your DNA fragments have been, how they have been separated, what are the particular base pairs when you compare it to the marker. Usually you can't see it when you just run the agarose gel. You need to stain it with a fluorescent dye, view it under UV light, and then be able to view the fragment lengths of the DNA that you want, the sample that you want, and compare it against a marker. Alright, so that's how it's actually done, practically done in the lab. Okay, but we've answered the question. It's ethidium bromide, just remember that. Okay, so we've come to the end of this video lecture, and I hope you've enjoyed today's video lecture and I hope you learned something new today. So don't forget to like, share and subscribe to MJ NEET Biology, and I'll see you in the next video lecture. And don't forget to follow me on Facebook, Twitter and Instagram for more updates. And yes, I'll see you in the next video lecture. Right, so bye.